All right, welcome to our monthly STEM seminar talk. Today we have Sunil Superman, and he is a professor with the Nutrition and Health Sciences Department at University of Nebraska-Lincoln. And today he's going to have a fascinating talk about taste receptors and the taste buds and beyond. So we're super excited to have him, and I hope you guys enjoy both here in person and all of you online as well. And if you have questions online, towards the end, we'll probably stop and maybe have some questions. You can also type in your questions, and then we can also ask those questions out to the audience. So
wonderful. Thanks, Stacey. So let's get started. Basically, thank you for having me for the invitation. Happy to talk to you all. So I will talk to you today primarily about taste receptors, and I've been working in the field for the last 10 to 15 years. This is pretty much the only thing I know. But before that, I also did some work with other senses. My PhD was in olfaction, and I did a brief postdoc fellowship on the sense of touch. So we'll see later on that these three senses are related. And up here you see these 5 senses, right? This classical classification from Aristotle, the 5 different senses: vision, taste, smell, hearing, and your sense of touch. But there definitely are more than 5, right? Because if you look at what is considered a sense of touch, that itself is, you can break it up into sensing heat or temperature, or touch, or pain, and so on. So that's already very multimodal. And then there's also the relatively new sensory system, that's the interoception, that receives the inputs from all internal organs, right? So, and also, and this is the system that we know the least about. However, it's definitely very important. Yeah, affect our physiology, our mood, and pretty much picking up. And sure, yeah, to the mood and physiology. Okay. So as I mentioned, I'm a taste and smell person. So what you mean by taste in everyday language is not what we mean by taste. So when we, for example, see a food and start eating it, practically all 5 of these senses are engaged, right? So for example, you see the food, so there's color in the food, the shape, and so on. And then once you maybe start eating it, for example, if it's a crunchy chip potato chips, there's the inputs of hearing to us that also contributes to our sense of taste. And then, of course, there is the taste coming from our tongue, taste buds of the tongue. And the nose is also engaged. And then there's also this touch pathway, that's the warmth, or the physical attributes of the food. Those are also engaged, right? But however, most of the senses, they come, most of what we call taste, they come from the chemosenses: taste, smell, and trigeminal system, right? So if you go to some restaurants, they dim their lights there, right? So while they're serving you food, why that is? Why they do that? Because when we lower our visual inputs, most of our brain is dedicated to processing the visual information. And when we lower that, so we kind of creating space for chemical senses, right? So basically, if you look at our evolutionary ancestors, they were probably nocturnal. So for them, both smell and taste and so on, and touch too. So the Roomba, the whiskers, and the mouse or the cat, they help them with navigating the surroundings. So those become enhanced without those situations. So what we'll do in this lecture is briefly go through smell of the trigeminal system, and then delve into taste, and then the post-oral sensory systems of the gut. So I have about 47 slides, so I'll go through them quickly and leave enough time for questions. So hopefully everyone run over time. So here is a diagram of the olfactory this is mouse. So right behind the nares is this olfactory epithelium, and here you have the olfactory septum, right? And then from here they project to this olfactory bulb in the brain, and from there it goes to higher brain region, right? So in case of smell, there are actually thousands of volatile chemicals that exist in nature, and now organic chemists are synthesizing even more. For example, synthetic fragrances and so on that they had to deodorants or uh, perfumes. So theoretically, we can make millions of mixtures from them. So if there are thousands of compounds, you can mix them in different ways to generate these combinations. And this paper came out in 2014, and there is some experiments, and they claim that humans can discriminate more than 1 trillion olfactory stimuli. So basically, that's a huge number. We don't know if it really approaches 1 million or not, 1 trillion or not. It's already beyond our everyday language. So whether we reach 1 trillion or, but it's definitely a lot, right? So our olfactory system is very, very capable. So the cells in the olfactory system, the cartoon of what olfactory epithelium. So there are basically 3 types of cells there. You can see these are the receptor neurons. These are neurons proper, and you can see their cilia, and that's where the events of smelling happen, and then the signal is transmitted to the olfactory bulb, and then over here, and then right from there, right? So, and then there also are some supporting cells which help the function of these neurons, and then there are also these basal cells which we now know are stem cells, because these neurons, they're exposed to everything. Right? They're exposed to microbes, they're exposed to harmful chemicals. So they don't live very long. Maybe 60 days is their half-life. So they are continuously regenerated from these basal cells. Actually, this is probably only one of the 2 populations of neurons that regenerate during our adult life. So if something happens to that, our sense of smell will weaken. So when COVID happened, especially these supporting cells were infected, and that kills a lot of olfactory sensing neurons and basal cells, and that kind of destroyed our sense of smell. So sense of smell of infected people. That is why the COVID causes. That's how COVID causes smell loss. So we saw that we can sense billions of compounds. So how does that work? So here the positive event is mediated by these genes called the olfactory receptors. It's actually the largest gene family in most mammals. For example, elephants have about 2,000 functional olfactory receptors and an equal number of non-functional ones. Now, cows have 1,000, the dogs have 800, mice have more than 1,000. Humans have less. Humans and primates have less, because now our visual system is more prominent. So we have kind of, we took our nose off of the ground or hand off of trees, right? So basically, our visual system has overtaken to some extent our olfactory system.
Can cows smell better than dogs then?
They can smell quite well, but you see here, dogs have 100 times more neurons. So it also is
they're more sensitive.
Yeah, it's also a function, not just the number of genes, but also the number of neurons. Right? They have large number of cells that are devoted to perceiving these senses. So that's one reason why they have very good senses. And see, this is a cartoon of the olfactory. Actually, not a cartoon. It's an electron micrograph of the olfactory, of the tip of the olfactory sense receptor neuron. You can see this cilia, and on this cilia you have this receptor. So these are probably the structure is familiar to many of you. These are the 7 transmembrane receptors, or the G protein coupled receptors, right? And so these were actually identified way back in 1991, and they're primarily found in the olfactory system. And as I said, the rodents have more than 1,000. We have about 400. So since we have a large number of neurons and large number of receptors, in combination, they can detect all these billions of compounds. So, receptor A might be sensitive to 10 compounds. Receptor B might be sensitive to 20 and partially overlap with receptor A. So that way, the system, a very complicated mechanism that decodes all this information. And so these are the two people who identified the receptors, and they also worked out along the collaborators, the organization and functional aspects of the olfactory system, for which they got the Nobel Prize, Physiology of Medicine. So, since then, we expand our studies to atypical model organisms. So sharks are another set of animals which have very acute sense of smell. Right? So actually, my PhD advisor started working on these animals, and what she found was that the sharks only have a handful of olfactory receptor genes. So it might well be that they have unconventional mechanisms for smell, meaning they might be able to smell only a small range of odorants, but what they can smell very well, because you see they have these huge olfactory bulbs, which are dedicated to processing the olfactory information. Right? And they also have a large number of so-called vomeronasal receptors. These are the guys which sense pheromones in mice. So, but maybe these receptors are now dedicated to sensing smell in sharks. So, using the same logic, we can try to guess how well a T-Rex could smell. So this is the brain of a T-Rex. It's quite explosive, but look at the olfactory bulb. They are very prominent. They are huge. So this is actually identified based on endocasts and scannings from the skull of T-Rex. So since that those olfactory bulbs are very small relative to the rest of the brain, so that probably means that they could smell very well. Okay, so then the second system is the trigeminal system. So this is part of the brain system. Trigeminal ganglion have all these the projections going to various parts of the head and oral cavity, and the ones in the oral cavity, they will contribute to sensing some chemicals and also the heat and the mouthfeel of the food. That's where that becomes important. And in case of the olfactory system, when we eat something and start chewing that food, those volatile chemicals that are released, they go back over to the nose, the nasal cavity, through the back of the mouth. And this is called retronasal olfaction. And actually, this pathway is more important for sensing taste of food once we eat it. Okay, so together, these two contribute a lot to sensing food. So we'll take a brief look at the trigeminal system. So in the trigeminal system, there are multiple kinds of receptors, but these so-called TRP or transient receptor potential channels, these are important for sensing many of the spices that we had. For example, this receptor called TRPA1 senses compounds in garlic, mainly allicin, and in cinnamon. Menthol is sensed by TRPM8, and this is also actually cold receptor. That's why menthol feels cold. Meat feels cold because it's kind of latching onto the receptor that is originally devoted to sensing cold temperature. So the mint feels cold. So camphor is through a related receptor called TRPV3, and chili, capsaicin in chili peppers, that goes through TRPV1, right? And TRPV1 also responds to hot heat, right? So that's why chili peppers kind of mimic that heat hot sensation. So the taste of chili peppers is that's hot. And then there is also the touch receptor.
I just want to let the people that is online know that we're working on the sound.
Okay, good. So are they able to hear, or is it?
They are having trouble hearing.
Okay, so I'll try to speak up a little more. Okay, so touch is mediated by these uh, uh, related channels, PSO1 and PSO2, and then of course TRPV1 and so on, right? So these discoveries made by primarily by these two scientists, they got the Nobel Prize in Physiology 2021 for those discoveries. So now we move to taste proper. So this is a cartoon showing the taste buds on the tongue. So you can see that make primarily three groups of taste papillae. These things actually you can see in a mirror, tiny photons on the surface of the tongue. On the front, they are called the fungiform papillae. On the sides, on the back, they are called foliate papillae. And on the very back, these are the circumvallate, right? And all these papillae have multiple taste buds. So here you can see taste buds arranged along this trench. These two papillae, then it is arranged on top. Right? So, and here is a cartoon of a taste buds. There is the taste receptor cells, which are packed together into an onion-shaped structure. There are about 50 to 100 cells in the taste bud. And the tips of these taste cells, they project out to the surface of the tongue through this taste pore. And then, very importantly, you have these nerves that take taste information from these taste buds to the brain. Okay? So all taste buds have these nerves and the taste pore. So the taste sense interact with these cells here, and then we have a transduction event, which then takes it to the food. So we now know what are the receptor proteins and genes that are involved in sensing this taste sense. For example, sweet taste is sensed by these two proteins together, T1R2 and T1R3. They join together, they heterodimerize to form the taste receptor. Sour, it was a mystery until recently, but now we know two channels that are involved in sour. These were very recent discoveries. Bitter taste, it's the middle like T2R receptors. Humans have about 25 of them. Mice have about 40. And amphibians, frogs, which eat lots of insects, which in turn eat lots of plants. They have about 100 or so of this bitter taste, right? So these actually are similar. Again, these are GPCRs. They have 7 transmembrane domain structure. Salty taste is still a mystery. We have at least one channel called epithelial sodium channel. There might be others too. And then there is umami, which is not so well recognized in all cultures, but that's real. One of the subunits of the sweet taste receptor, T1R3, is also involved in umami taste, but in this case, it joins up with other receptor called T1R1 to form that umami. So our sense of taste is actually highly valuable across evolutionary timescales. So cats, members of the cat family, which do not eat any plant food, right? They have lost their T1R2 subunit, which is involved in sweet, right? While giant panda, which only eats plants, like highly restrictive diet, they do not see a lot of proteins in their diet, so they have lost their T1R1. They can still sense sweet. Cat still can sense umami. Cat food manufacturers, they have a hard time deciding the right food for cats because they like eating tuna, but you have to make sure that adding sugar to their food is not one of it. So, but in our case, we have all the receptors intact. Our sense of taste is highly developed, so we can enjoy a wide variety. So, you might have seen this diagram showing that different regions of the tongue are differentially sensitive to very different types of taste sense. So glitter was supposed to be bad, sweet in the front and so on. However, this is not right. This has now been thoroughly debunked. There's an interesting story behind how this came about. So the original paper was published in German. It didn't claim anything like this. There are some subtle differences between different regions of the tongue. nothing as dramatic as this, but when it was translated into English, they didn't do a good job. So this was way back in 1980. 1880 was when the publication came out in German. 1945 was when it was translated to English. And since then, you see this everywhere. So my son got his first science textbook, and there it is. But we don't have it. One will come out here.
I'm gonna try with these microphones.
Okay.
Figure out.
Well, the sound maybe.
Yeah, let's see if it works better.
So see how that work. Okay. Like,
Only give the projectors, like, you have to find a place that's outside of the, because it shines in your eyes.
yeah, that's right. So
Yeah.
this is good. So
Okay.
probably I'll try.
Okay.
So that's a tongue map. That's totally bogus. So and here is the classification of taste cells. You have multiple kinds of taste cells. There's some that respond to bitter, some that respond to sour, salty, and so on, right? And they express only that particular kind of receptor. Sweet cells will express only sweet taste receptors. Bitter cells will express only bitter taste receptors, and so on. And just like in the nose, we have stem cells which are not shown here, but again, these cells they do not live long. They live maybe a month or so, and then they die off. So they have to be replaced by the stem cells population. And we know several genes that are expressed in these cell types, including TAS1, TAS1, etc. What that we met a little bit. Also, so we'll go through sweet, bitter, and umami taste a little bit. We won't talk any more about sour and salty. So sweet taste receptor, very important receptor, right? Helps us enjoy the sweet taste. So as I mentioned, you have the two subunits, T1R2 and T1R3, and they join together. And some of these studies, multiple studies carried over in many labs, including that of my advisor. They helped map where all these sweet taste ligands bind. And so here is, for example, in this region, you have sucrose and fructose, right? Binding there and so on. So, and here, these are caloric sugars, right? Carbohydrates. And these are unrelated chemicals, right? These are sweetness enhancers that were discovered recently, which by themselves do not have any sweet taste. But when you add them with a little bit of sucrose, the sweetness of sucrose is enhanced. So this discovery came about maybe 10, 15 years back. And you might recognize some of these artificial sweetness too. This is from stevia, Reb Reb C, and so on. Here is a few others, cyclamate. I forget which one it is part of. You have probably Splenda. And there are also some sweet proteins. Right? So there's a wide range of substances that can taste sweet. And we also have some compounds which can keep it sweet taste. So that these are actually very popular in some cultures. So gourmarin, or the name means sweet slayer. So if you consume gourmarin, your sweet taste will be knocked out for half an hour or so. Yeah. So basically, your appetite will go down. So there you can purchase this gourmarin tea from Amazon. It works very well. So we have a Discovery Day in our East Campus where we have people come around and we have a stall there. So we give them gourmarin to taste, and then we give them sugar before and after, and the difference is pretty strong. It tastes like, after you consume gourmarin, it tastes like sugar tastes like sand. Just the texture is left, taste is gone, but it'll come back after half an hour. But it's very good for reducing your appetite, so you won't overeat. So it's very popular in India, in Japan, so it's also a very good anti-diabetic for that reason. Okay. So there is some myth busting required for sugars and high fructose corn syrup. So we'll try to do that in the next couple of slides. So when we say sugar, we typically mean sucrose, right? Table sugar. This is produced by many plants and some cyanobacteria. Fruits are rich in that. So you have basically glucose and fructose joined together to form your sucrose, right? And then we have high fructose corn syrup. So what that is and why that is kind of controversial, we'll try to see that. So basically, corn syrup is made from corn, and most of it is 90% of it is glucose, right? And so, however, we convert corn syrup to high fructose corn syrup. So why do we do that? The reason is, you can see that here, glucose is the least sweet, sucrose is the next sweetest and sweeter. Sucrose is sweeter and fructose is sweetest, right? So glucose by itself, glucose solution by itself, is not very appetitive. But if you convert some of that glucose to fructose, then it becomes, very sweet, right? So basically, what you do to make high fructose corn syrup, you take cornstarch, and then you break it down to glucose using amylase, typically isolated from some bacteria, and then that glucose is converted to fructose. Not all of it, but at least 50% of it is converted to fructose using a second enzyme, cellulose isomerase. So now you have a solution. So sucrose is 50% glucose fructose, and there are two grades of two formulations of high fructose corn syrup. HFCS 55, which is 55% fructose, and 42, which is 42% fructose, right? So there's a lot of controversy surrounding high fructose corn syrup, and there have been lots of studies done on that. And the conclusion of all those studies is that it appears unlikely that high fructose corn syrup contributes more to obesity or other conditions than sucrose. So basically, the reason why we have a scare surrounding high fructose corn syrup is countries which had a very bad obesity problem, primarily US, they started using HFCS, maybe heavily around 70s and 80s, and that's also when the obesity epidemic really took off. So it's basically a correlation between these two, but as you know, correlation doesn't mean causation. So it's just that people were consuming a lot of high fructose corn syrup. So if you consume a similar amount of sugar, again, you will have similar problems. So there's actually no big difference between these two, right? So when we eat sugar, obviously it's a disaccharide, but immediately as it gets into the gut, it's broken down into glucose and fructose and then absorbed by the body. And then there are the artificial sweeteners. Many of us take them. Some of them were developed from sucrose. For example, sucralose from Splenda. It's a chlorinated form of sucrose. But now we have many more in the market. Struvia is from stevia. You have equal and so on. And so about 6 to 7 of these artificial sweeteners are approved for human use, and you can purchase them. So they do not have any energy because they cannot be broken down and metabolized, and we only need small amounts. Many of them are hundreds to thousands times more sweeter compared to sucrose. Okay, so now we come to umami, and umami is the flavor of amino acids. Humans are sensitive primarily to glutamate and aspartate. So basically, the H is the ionized form, right, of an amino acid. So glutamic acid means H is glutamate and so on. So if you want to add glutamate to the diet, you have to create a salt form of it. Basically, that's more stable. So basically, you react glutamic acid with sodium hydroxide, and then you get monosodium glutamate, right? So there is a lot of health scare surrounding MSG consumption, and which we now know is not a concern because what is this basically it's glutamic acid, one of the 20 amino acids in the proteins, right? And then sodium. So if you eat any protein source or salt, you will get both of these. You have to really consume buckets of it to have health issues. But actually, this is a very subtle taste for many people, but it's actually adds a lot to the flavor. So there's no need to be worried about MSG either. Okay, so that's all about umami taste. So now we come to the bitter. So bitter taste. This is typically the aversive taste, right? Classically we think of bitter as something that can poison you, right? However, there are thousands of bitter compounds, many of them being beneficial. And these include coffee and practically all medicines are bitter too. So, and as we mentioned, the receptors for bitter are the T2Rs, right? So you have about 25 T2Rs for humans. So in combination, they can sense thousands of compounds. And and as you mentioned before, sweet, bitter, and umami receptors are GPCRs, right? So you have these receptors, and they share a downstream signaling pathway. All these components that we see here, once a ligand binds to that receptor, it produces a signal through this signaling pathway. So each step of this pathway, the signal is amplified, and then finally, you have the release of this neurotransmitter called ATP, which is actually more popularly known as an energy source, right? But ATP is also a very common neurotransmitter, and that signals to the taste nerves, and then that information goes to the brain. So at this point, I don't want to miss the chance to sing the virtues of GPCR. So these are actually a very important class of receptors. They are sensitive to multiple things, not just taste and smell, but for example, the receptor for regulating our blood pressure, the angiotensin receptor is a GPCR. The beta blockers bind to GPCRs. So, they're very popular drug targets. The reason for that is because they are sitting on the membrane, accessible to whatever drugs or chemicals we take in, right? And they're also important in regulating many of the bodily functions. So about 57% of drugs in the market target GPCRs. So very important class of receptors. So, I don't know if you have heard of this, super tasters. Some people are supposed to have, better ability to taste, and these are called the super tasters. And there's one theory that they have more taste papillae, and by extension, more taste buds in their mouth. So here they have stained the tip of the tongue with a dye that will help us identify the taste papillae. So the theory goes that people who cannot taste so well have fewer taste papillae compared to normal tasters, and then super tasters have even more. So if you have more taste buds, obviously that might mean that you have a higher ability to taste. So, but it's still a theory at present. Maybe it needs more work. So this is until now we have spoken about events that happen in the oral cavity, right? Once we ingest some food and chew it. However, sensing continues after we swallow our food. So our digestive system is involved in both absorbing that food. The gut is involved in both absorbing the food and sensing it. So we'll look at that sensory function and then see how discoveries around that help us develop the GLP-1 mimetics. Right? So here, these are some experiments first done in the 1960s. So here what they have done is they have given a glucose solution through two routes. One is oral, right? And then the other is intravenous. So if you track the blood glucose levels after either IV or oral glucose consumption, there is no significant difference between these two conditions, right? Because we are basically adding in the same amount of glucose. So it will be initially the glucose levels will rise, and then our cells in our body will absorb some of the glucose, and so with time the glucose levels will come back down to baseline, right? But if you track the insulin responses at the same time, you see that huge difference. So IV, it does trigger an insulin response. There is definitely an elevation in the insulin level. However, when we take it to the oral route, there is maybe a 2 or 3-fold more elevation in that insulin level. So, right. So, what is the implication? Implication probably is that there are sensors in the mouth and the gut which senses glucose, right? And that contributes to triggering an insulin secretion, right? So obviously the pancreas can sense glucose and secrete insulin by itself. However, there is something in the mouth and the gut that is priming that insulin secretion, right? So they didn't know what this is. So they just call these incretins. These are supposed to be some peptide hormones that were released by the gut and the mouth in response to glucose, right? So, so where do these incretins come from? So this is a cartoon showing the intestinal epithelium with the various kinds of cells in that. So you have the crypt where you have stem cells, and then some cells that are generated by stem cells but are still not mature intestinal epithelial cell types. You also have cells that secrete mucus called goblet cells, and most of it is actually the enterocytes which absorb nutrients. And then we have this other cell type called the enteroendocrine cell, right? So we can see them here. These are very few compared to the enterocytes. Actually, it's this here. These are transit amplifiers. So these are the enterocytes here. So, and what do? So they are the ones that secrete this hormone. So basically, when we eat some food, they're digested to the monomers, right? So starch is digested to glucose, triacylglycerols are digested to fatty acids, and then proteins to peptides and amino acids. Right? So we have the digestive enzymes in our gut that do this, and then our body absorbs them. However, the absorption of these carbohydrates and fatty acids and so on, that is coupled to nutrient sensing, and that is where the enteroendocrine cells come in. So this is the electromicrograph of an enterocyte, and you see they absorb all this food. But interspersed with these enterocytes, you have the enteroendocrine cells, right? So here you can see that they have the receptors. So the sweet taste receptor, T1R2 plus T1R3, plus another receptor called SGLT1. They sense carbohydrates in enterocytes. Amino acids, you have T1R1 plus 3, which we met for umami taste. They are also present on enterocytes. There are receptors for fatty acids as well, right? So all of this is sensed by these enterocytes, and then they secrete a bunch of hormones, right? So we have GLP-1, GIP, CCK, and so on. That's involved in satiety. Some others like motilin and the serotonin, which help the motility of the gut and so on. So basically, the enteroendocrine cells, they're distributed throughout the epithelial cells in our gut. Stomach produces ghrelin, which is the hunger hormone you might have heard of. And then there are various enteroendocrine cell populations within the small intestine and large intestine. For example, somatostatin comes from the so-called D cells. CCK or cholecystokinin comes from I cells. L cells produce GLP-1, GLP-2, and so on. And then you have 5-HT from enterochromaffin cells and so on. Right? So actually, our gut surface is very big, right? So although the enteroendocrine cells represent only about 1% of the cells in our epithelium, by their sheer number, they constitute the largest endocrine system in the body, right? So, and they secrete this bunch of hormones. And how do these hormones affect our physiology? There are two routes. One is through the blood, right? So they get secreted to the blood, and then the blood takes those hormones to various parts of the body. And then there is this nerve called the vagus nerve, which innervates our gut, and they have receptors for these hormones. And then so the vagus nerve takes that information to the brain directly, right? But through the blood, it can reach practically any cell, any organ in our body. So that becomes important when we discuss the effects of this hormone. So initially, actually the gut nutrient sensing has two effects. So one, in the initial phases of feeding, is that it promotes appetite, right? So I don't know if you have noticed it. So we might eat some food, you might not eat the taste, but after some time you might want to eat more of it, right? So, and that might be coming from this system that's engaged first, that promotes appetite. So again, in this system also, you have these various receptors for sweet, including T1R2 plus T1R3 fats and proteins and so on being engaged. So this is the one that this system is what gets engaged initially during feeding. But however, later on, once we have eaten sufficient amounts of food, all these other hormones, GLP-1, CCK, PYY, they are secreted from the gut, and then they are the ones that promote satiety, right? So they give us the feeling of full.
You guys plan on taking about sound? Is it still acting up?
Ask. All right. How do you feel about the sounds people online? Sorry for the interruption. Don't worry.
Issue.
Bye.
Oh, so where's is that running the Zoom meeting or is this right? Okay.
Sound on fire.
No.
We can probably try to speak it from there.
Just all the start stuff. Yes, it was not.
There's audience is mentioning that they are feeling that is working good now.
The thing is working good now.
I'll say it's working now.
Okay, I just
Probably-
got up to count.
Okay, that's in the trick.
Don't take this down. I'm having a hard time hearing too. With it possible to like judge? I'm having a hard time hearing too.
Thank you. So let me try speaking from here now.
Okay.
Are they able to hear better?
Yes, but like in the middle, wait, what? I bug you if I'm moving the mouse. I think that I'm happy, but I don't know. Like you tell me.
Yeah, so I think it goes to all the screens, right? So let me try. Is that better now?
Can I hear him?
Okay, great. So all these hormones that we just saw, GLP-1 being the most well-known, but you have these all others, right? So they get produced later on once we have eaten sufficient amounts of food, and they are the ones that promote satiety, right? That's not.
Not working.
Yeah, that. Okay. So the most popular, well-known among these gut hormones is GLP-1, right? So we'll focus on that. It's a hormone that was discovered way back in 1980, and what we now know is that it regulates pretty much every organ in the body. Right? So we have receptors for GLP-1 in the adipose tissue, in the kidney, in the muscles, in the bone, in the stomach, in the brain, pretty much all tissues. So remember, it can go through the blood, right? Like a classical hormone. So when we eat something, GLP-1 can be released and get to pretty much all parts of our body, right? So, the GLP-1 mimetics, they are very helpful in promoting satiety and preventing us from overeating. However, it turns out that they are useful for other things too, and that reason might be because the receptors for GLP-1 are found in pretty much all cells of many organs of our body, and then so that way it can regulate the function of pretty much or a large number of organs, right? So usually native GLP-1 produced from our body is degraded by this enzyme called DPP or dipeptyl peptidases. So they have very short half-life. So within a minute or two of them being released to the blood, they start getting chewed up by this enzyme, and so the level of this GLP-1 and GIP goes down very quickly after feeding, and that's what we want. So we don't want to be sated all the time. So initially after feeding, we want to be sated, but we want our hunger to come back. But if you want to use them as a drug, that's a problem, right? So now they have designed many GLP-1 receptor agonists. These are synthetic molecules that mimic GLP-1. So here is the site in GLP-1, which is subject to proteolysis by this enzyme. So in all these GLP-1 mimetics, this site is mutated. So here is an alanine in the native GLP-1, here it is glycine. And in this case, it is conjugated to albumin to protect it. In case of semaglutide, you have I think that's it's not as it's arginine, right? So you switch the amino acid around, so that means that this protease cannot chew it up anymore. It has been linked to albumin, but using biogenetic engineering, it's linked to antibodies and so on. So you might have heard of all these compounds that are now part of Wegovy or whatever injectable GLP-1 mimetics you can get. So initially, when they made this GLP-1 agonists, they were made to treat diabetes. However, that was approved, I think, in early 2000s. And in early 2010s, they found out that they are very good at promoting weight loss. So they promote satiety, and then by promoting satiety and so on, they reduce the amount of food that we consume, and then it's good for treating obesity as well. So initially, when these findings came out, the share prices of Walmart and the Pepsi, Coca-Cola, they fell because it was thought that people are not going to snack and consume beverages anymore. It's not as much, because if they get access to drugs, then might be an issue. But the promise seems to be holding up so far, right? So they become very popular. About 10% of the population in the world now take this GLP-1 mimetics. And what's interesting is that they're now being found to be effective for treating ailments of kidney, liver, heart, even mood and sleep disorders and so on. So basically, as I said, these receptors are found in very many of our organs, and that way they can regulate various aspects of our body, right? So and this receptor again is a GPCR. So you realize how important. Okay, so that's all I have about nutrient sensing. However, I have a couple of slides on the opportunities for students here. So we got funded for a grant to study the roles of zinc in taste cells. So zinc maybe is not so well known, but it's actually a key micronutrient. Very important. About 10% of the proteins in human proteome are regulated by zinc. It can regulate our growth and development, immune system, nervous system, reproduction, and then it helps to translate and transcribe our genetic information and so on. So it's a very important micronutrient. And what we also know is that it has some special roles in it. So here is the taste papillae of a mice that on the left side, you can see papillae of mice that are fed normal diet. And here is the taste papillae of a mice that was fed a zinc deficient diet. You can see the taste buds are atrophied in this case. They don't develop well. Okay, but here, and actually, their ability to taste also goes down. So here you can see the ability of the mice to sense sodium chloride. It's affected in this mice, and actually, when somebody goes to the doctor with the taste loss, and doctor don't know what's going on, the first treatment is to give a zinc supplement, and that will help cure that taste deficiency in many of those people. So, so we don't know how that works. So that's we got some funding to find that out. So we'll use mouse models for that. And the second role for zinc is in sweet taste. So zinc, if you mix it with any sugar or artificial sweetener, it inhibits the sweet taste by about 70%. So we don't know why that happens. So we have some theories, mostly around the zinc transporters that transport zinc into our body cells. So so that is something that we want to study. So we just got this new grant. So we're very excited. And so here's that part about it. Quite strong those we can see that you had various concentrations, right? So because we see sulfate, it goes not a lot, but magnesium sulfate pretty much similar to glucosanol, right? It's not the sulfate part, it's the zinc part or the magnesium that's doing that thing that's doing. So you want to know why. So for that, we have some internships for students. So we are looking forward to some of you joining our lab summer. I had doing some work there. So if you're interested, please reach out to your coordinators here. We are in touch and also can reach out directly to me. But typically, we want to start these internships in June, and we want to go. That will go for 2 months. So it's a paid internship. So hopefully, they come to our lab, help us, and learn some stuff.
Usually these are like, I think like 10 weeks, right? Over the summer.
Yeah.
And then it's usually like 40 hours a week for 10 weeks. Yeah. And fully paid. Yeah.
Hopefully, some of you will consider this opportunity. And that is all I have for now. Some acknowledgements that we have been.
Ask questions now. Yeah. Sorry. All right.
Let's do this for my
There's gonna be a camera on me.
You have a question?
Oh, sorry. I
Yeah.
didn't know if that was good. I going back like several slides ago, there was like you mentioned that like only like certain ion channels on the nerve detect certain flavors. Like some detected like cinnamon, some only detected like chili and all that stuff. Are you saying that like certain parts of the nerve will detect certain tastes or like certain flavors?
Well, not certain parts of the nerve, but all these nerves originate in our so-called somatosensory system, right? So from the trigeminal nerve, there are subpopulations of nerves which express TRPA1, which is a sensor for heat or capsaicin, right? So that is when you excite that nerve, that is decoded by the brain as a hot taste or hot temperature, right? So, and then we have a different set of nerves which will express TRPM8, and they will not express TRPV1, right? So if you excite those nerves, you feel a sensation of cold, right? So these are segregated pathways, right? So it's different sets of neurons within the same ganglia, some responding to heat, for example, others to cold, right? So that way the brain knows how to decode it, right? So if neuron A that expresses TRPM8 is excited, you interpret that as cold, right? So now when you take menthol, which is also able to bind that TRPM8, right? So then you perceive menthol as cold, right? So you're chewing gum with chewing gum, you perceive it as cold, but it's actually not cold, right? But it's just mimicking that sensation because you're able to excite those cold sensitive neurons, which express that cold sensitive receptor, right? That logic goes to large kinds of neurons in our sensory system. So for sweet, we have sweet sensitive neurons which pair up with sweet sensitive taste cells. So when you excite that sweet taste receptor, that goes along that pathway. The neurobiologists call that a label line. So you just have to excite that particular receptor, right? Either with, in the case of the good example is capsaicin and menthol, right? So they are not hot or cold, but since they are able to activate that hot or cold receptor, their taste mimics that hot or cold sensation.
What about different people's different tolerances to like hot flavors? Like I'm a baby about spicy, but some people can hit it really hard and feel like great.
Well, that's a also it's partly a question of your food experience with food, right? So if you're brought up in a culture that eats a lot of chili, then obviously you will
Used to it.
get used to it. So you get desensitized to some extent. But one theory about sweet, people who are sweet tooth, they might have a less sensitive sweet taste receptor because of some polymorphism receptor. So support them to get that sweet kick. You need to consume more, right? So that's one theory that
Have they found any
they have
polymorphisms?
very polymorphic. In fact,
I was
bitter.
scream myself.
Yeah, bitter. Bitter is actually a fertile ground for polymorphism. So bitter taste receptors and also smell receptors, they have a large number of polymorphisms. These are the genes which have a lot of polymorphisms in the population. So you might have a set of, so remember there are 25 of these bitter receptors and 400 of smell receptors, right? And then there are this large number of polymorphisms in the population. So you might get a certain set of alleles for your smell and bitter receptors, and your sibling might get a slightly different. So you might like a particular kind of receptor, sibling might not, right? So that kind of population.
But there's no way like you can have an upregulation of the amount of receptors.
Yeah, that's possible.
That is also possible.
Yeah. So there were trials done in my previous institution. So it works also for salt. So you cannot reduce sugar in one go. Right? So what you do is the people were enrolled in a trial. They were given soup with each week they reduced the amount of sugar or salt in that by a little bit, and by week 6 or week 8, they could consume maybe let's say soup with half the amount of salt that you would find in a typical that you purchase supermarket, right? And still find it palatable, right? And when you look at the receptor expression, it has gone up. What was sweet?
So you become more sensitive when you get less.
That's the way to
Yeah.
reduce our overconsumption of salt or sweet. But obviously, our food environment is so bad. You go to any bakery section of supermarket, everything has load for sugar. It's hard to practice that, but if you're able to control.
That's interesting.
What about with age? Does it change something like I have ages?
Yeah, that's an excellent question. So actually, kids are very sensitive to bitter, especially toddlers, right? So there are some explanations for that. Basically, it's at age 3 or 4 is when we leave your mother a little bit and try to explore your surroundings and try to explore the food surroundings. So what does that mean for a kid? Before safe foods were not available, right? During
Of the forest.
yeah, but you have to be very picky about what you eat, right? So today I have a very heightened sense of bitter. That means that you become very picky, right? So that helps in our pre-agricultural societies. But now, most of the food that we give to our kids are safe. However, since they are sensitive to bitter, they become very picky. They don't eat vegetables, right? And on the other hand, when we become old, the regeneration of both our olfactory neurons, the smell neurons, and taste cells, they slows down, and our ability to sense taste goes down. So
I mean, I wonder if that's like sometimes older people just don't eat as much, or
that's right. So when another thing is when you do some chemotherapy for cancer, so all these agents, you lose hair because it's like dividing cells, but we also lose our space cells. That's a huge problem for that population because they don't eat because food tastes like cardboard for them. So that their appetite goes down.
so you need to find some sort of way to upregulate. Like, can you? Yeah.
But at least help them to recover that regeneration quicker so that the feeding is so actually muscle wasting is probably the biggest reason why chemotherapy, or one of the biggest reasons why chemotherapy fails. If you don't eat well, you cannot able to recover, right? Sure.
What was going on with people losing their taste with COVID?
Yeah, so most of that was actually people losing smell. So those or the carotid epithelium, right? The supporting cells which get infected by COVID, and then release all these inflammation genes, and then that destroys both stem cells and neurons. It also happens in taste buds. So that means they are killing that off. But fortunately for most people, it comes back. But for many people, it didn't come back well, or when it came back, it became distorted.
Sunil, do you want to come talk here in the middle? Because I think
know.
I think the people online, I think these speakers in the middle, we need to adjust this, but these speakers in the middle are picking up. They can hear me, but they can't hear you. So if you come,
Okay.
it's a little bit awkward, but just come here in the middle. Yeah.
Okay.
There's a question in the chat, and it says, how does our brain know what taste is good and what taste is bad? For example, why some people find olives tasting good and some find them tasting bad.
Yeah, that's a good question. So some of this is innate, right? So sweet, for example, is innately appetitive, right? So you have that, neural pathways for sweet are they are tuned to they develop by development itself that's for appetite. It's triggering appetition, right? It's triggering a preferential taste sensation. However, we talked about the polymorphisms, right? So we have different variations in the population among both our smell receptors and bitter taste receptors, right? So that means that person A might be able to sense some components of olive, for example, and then if it is triggering a bitter taste, then they will find it aversive. But somebody else might have a slightly different version of that receptor, which is probably not as sensitive to that bitter component, right? So that means they won't find it as bitter. So, but however,
Sorry. So
one last point I want to add there: our preference for food is also shaped by our experience, right? So,
Like your culture, your
yeah,
environment.
so if it is all by taste, nobody will be able to be drinking coffee, right? So, but we know that's not true because coffee, once it gets into our body, it has so many other positives. So you have.
Oh, so is that why the people are like more influenced to continue, like when you're adapting a palate for certain things. So like when you're drinking coffee, you're saying you have certain receptors that pick up on certain things. What about like, let's say here where at first you get these very abrasive flavors and, sensations, but then you kind of brought that up. You just, it's like you adapt to it, or so how do you explain the
Yeah, so it's not exactly adapt. So what we call this is the post-oral effects of nutrients, right? So once it leaves the mouth, our body absorbs it. So coffee triggers our adenosine receptors in the brain, and that causes some level of arousal. So our mind becomes sharper for a while. So we like that. We learned that, right? So there's always that learning component going on. Once we consume a food, and then after several hours, the full effects of that plays out, and then you learn to like something. Not
Can
like-
you explain then why, like, is it just such a powerful negative reaction when they first start having something like that it just completely negates like positive that comes from it? Because like when you drink, like you have a glass of wine, you do. I swear it's like it opens up and you all of a sudden you start tasting new flavors.
Yeah, sure.
Yeah, so it's hard for me to wrap my mind around.
Yeah, so it's drinking something is a multi-sensory experience, right? So something sensory, the conscious sensory effects are from the mouth, right? But once we swallow it, then we absorb it, and then it does its own set of things, right? So, like example, for coffee being causing arousal, which is a positive experience for us, right? So then we now learn to consume coffee, right? So same thing probably goes for wine, although not all the effects of wine are positive, but it also gives a certain arousal or disinhibition of isops, which makes us happy for a while. Right? So we learned that. So all that learning is also part of our liking of food all the long, right?
Wait, I have a weird futuristic question. So, what if we could profile people's polymorphisms and like have a profile of taste and then like make food that's like designed for their taste and they can have like a crazy experience?
So actually people have been trying to do that. So they've tried to type the polymorphisms for their smell and bitter taste receptors, and then try to find out using food frequency questionnaires whether that polymorphism is linked to their eating habit. So there is definitely some good correlation there. However, again, the post-oral effects is coming.
Yeah.
So even if it is not a preferred food initially, if it's good for our body, once it is distributed all over, then the post-oral effects kick in. Then obviously we might learn to like that food. So it's not very clean. So because there are multiple components to eat. But there are some interesting things. For example, patients, there are kids born with AIDS, which they get from their mother, right? And for them to consume their medicine. So you cannot give a tablet or a pill to a kid. It has to be a solution, right? So, and they're incredibly bitter. And one of the projects that I worked on briefly was to identify something that will block that bitter taste, so that those kids can
Take the medicine. Yeah.
Yeah. So with that, would that be related to why some people can smell or taste asparagus while others can't?
Yeah, I think asparagus and
Cilantro?
cilantro.
Yeah.
Cilantro. That's
People are like, they think it tastes like
we
nothing.
don't know which gene it is actually. That gene of cilantro. That's the smell receptor.
And question over here.
Sorry, no, this is a good conversation though. I like this, but like I did have a question about like fructose. You said that fructose was like the sweetest among the simple sugars, right? Do you think there might have been like an evolutionary advantage as to why we have adapted to eating like detecting fructose or sweeter, like rather than like glucose or what was it, maltose? No.
Well, glucose is all obviously it's in the grain, for example, glucose. But in fruit, once it ripens, you have enzymes there that convert that glucose to sucrose, right? So that turns out to be better ligand for our sweet taste receptor, and that's a case of co-evolution between plants and animals, right? So plants cannot move around, so they cannot disperse their seeds. But if you pack their seeds with the fruit, and that way entice animals to eat them, a day later, that seed get deposited somewhere else in the forest along with a pile of fertilizer, right? But then you get that seedling has a very good start in life. So it's a case of co-evolution between plant and animal. So,
Like thinking it's like a chicken and an egg situation, but like
yeah.
who's first? Like
So you can think of it going back in the flowers. Why do we have nectar, right? So it's to attract the insects, right? So the plant trades some of that energy in honey or the fruit for it to be able to disperse its seeds, right?
so is it like we already inherently detected fructose as sweeter and then the plants adapted to that? Like
Yeah.
okay, got
So
you.
yeah. So this is when did angiosperms originate? Maybe 100 million years back, right? So since then it has been going on. There's been a lot of time for this to play out, right? So yeah.
Interesting. Okay. I guess I had another question about like how we can like train ourselves to like certain flavors more as we expose ourselves. I kind of like the good point where it's like we kind of train ourselves to like beer and wines and start to detect those like more subtle flavors. Does that start in like training like the mouth or like does our brain like
Yeah, the
think about it?
so in case of liking foods that have inherent bad taste or bitter taste, that's of most of it comes from the so-called post-oral effects, right? So various parts of our body processing that nutrient, and then that plays out in our body, and then if it's good for us, we will learn to consume that food more, right? So
I guess I was like, sorry, I should make that more clear. Like, are there like physical changes in our taste buds at all when we try to like,
but there might be some desensitized. Mostly it's the brain, yeah.
mostly takes them?
Yeah. So, but if you have a very sweet diet, actually brings down the amount of sweetness receptor in your mouth, for example. You become less sensitive for that because the gene expression for that receptor is turned down a little.
Like a feedback loop here now. Like if that's what I was trying to get, because it's like there's like this abrasive, it's almost traumatic when you at first when you're like going at it, and then but after a while,
Acquired taste because the after things very right. It's bad.
right?
No, you say good for the body. I'm thinking, yeah, like people getting, yeah, they're just happy, right? You're like, woo, this is the best experience. Yeah. I love this wine. The other question.
Is there a correlation between like the hypo tasters, super tasters with like pickiness?
Yeah, it's a good question. So it's still a theory, has not been tested very well, but hyper tasters, super tasters, they're supposed to be more picky. They don't drink a lot. So I don't know. If you go to many.
Definitely have the taste test things.
Uh-huh.
My purse. It's like a fun learning trick.
Yeah. I mean, if you go to some science fair, they will test it out for you and tell you that you are a hyper taster or a super taster or a normal taster. I've seen that. I mean, it's still a little bit iffy, but we do see those differences in number of taste papillae. So as somebody who studies taste regeneration, that part is very interesting to me. But no, we don't have good models to study. So probably want to look at it now.
So would you think that somebody who is, let's say, an executive chef of like 5-star Michelin restaurants or whatever, would you say that they would probably be like, would you say they'd be a super taster or not?
I mean, if they're super taste, they become very picky.
Right.
So the theory is super tasters are picky because they have heightened sensitivity for everything, right? So probably they are normal tasters or hypo tasters, is my guess, if this theory is true.
Yeah.
I mean, they are very picky. The people who don't drink alcohol, they are super tasters.
We are, you're saying?
Typically, yeah. So you can.
Fascinating.
Thing is also as we get older, things change, right? So it's all a interaction between these various biological things, and later on, high age and so on. So yeah, we know. Yeah. It's not very clean in real life. Right? So you have one more.
Yes. So back to the graph that you showed earlier about like the oral versus IV insulin spike. Do those like same sugar sensors that you mentioned in like the gut and mouth, would they have a similar like, reaction to artificial sweeteners and like also cause a insulin
Yeah.
spike?
So yeah, no, actually, thing is, so I showed the two receptors, right? T1R2 plus T1R3 for sweet, for example, and that will be sensitive to both artificial and caloric sugars, right? However, SGLT is a sodium glucose co-transporter. So this is an interesting receptor too. It's actually also a transporter. It co-transports glucose along with sodium. So when sodium comes in, that causes a depolarization. So you remember the way that nerve impulse is transmitted. So that is only sensitive to caloric sugars. Right? So you have one system that can respond to both caloric and non-caloric sugars, and another one that's now responding only to caloric sugars. So it's a sum of these two. And actually, SGLT1 is more important in the gut than in the mouth. So almost that gut sensation depends a lot more on having those glucose there as opposed to something like aspartame or stevia. So yeah.
Stevia, though, doesn't that actually have a number of carbohydrate? I think it's stevia. Like there's a stevia out there that has a level of carbohydrates, but not enough calories.
Yeah, small amount of sugar, like glucose or sucrose, but the most of it is actually filler. If you open a pack of artificial sweetener, maybe 95% is filler, 1 or 2% is your sugar, or 3 or 4, and then a tiny amount of non-caloric sweetener. So if you go to Europe, right, so they have like really minute tablets. You can even see them or the sugars. They don't add fillers that much. Here we add a ton of filler. So I think they add maltose or maltodextrin. That's a filler. So that just to give that bulk to that, right? So
Like feel like you're adding something.
yeah, you feel like you're
Like this is so much. It's gonna be so good,
yeah.
and it's like a little bit.
Very tight. So they are actually aspartame is like 1,000 times sweeter than sugar, right? So, and some of them newer ones are 10,000, and the proteins are 100,000 times more sweeter. So if you find a way to add them to baked goods, that so
Yeah, because like the packets for sure and all the artificial sweeteners are the same size. So yeah.
Probably did it on purpose. Like you're getting the same thing.
yeah.
They are quite different, and they're quite different. Like I use cereal, both
Right.
almond.
I feel like
Yeah, no,
the same amount.
they may, yeah, probably, yeah.
They're sweeter.
What Hugh was saying though, okay, so this is something that says National Institute of Health has even discovered that there is premature release of insulin, which could lead to insulin resistance, even in non-polar sweeteners. So isn't that what we're kind of talking about there?
Yeah,
That's what
actually gut has the T1R1, T1R3, pancreas has that, hypothalamus has that. So we never know. Yeah.
Is it the mouth though? You know what I mean? Like, you know, how we were talking about, like when you intake sweetener or sugar, that's when the insulin process starts, right? It's not like quiet.
Yeah, that's a cephalic phase. So the bell ringing and salivating. That that thing is called cephalic phase, right? So, and some of that is also linked to secretion of digestive juices, insulin secretion, right? So that's cephalic phase. So yeah, so but the effects of this non-caloric sweetness might be related to how they overall affect our feeding behavior. So you get a sweet sensation, but you're not getting calories. So you are going to your cafeteria, taking a bottle of zero, Coke Zero, whatever. You drink that, you get a sugar kick, right? But then you're not getting calories that usually are paired with that. So you might end up eating more of your food because to compensate. So,
I did have one more question though. So I know you were mentioning about how zinc deficiency makes you like lose your sense of taste and smell. Isn't the opposite true where like if you have too much zinc, you have like a metallic taste in your mouth and you like kind of kind of like blunts your taste of
Yeah, so zinc we don't usually consume that much, right? These are micronutrients. These are like consumed in micrograms. So normally there are no foods that contain so much zinc that it will feel metallic in your mouth. So it's like really tiny component of our diet. So, but I can assume probably you, if you add a lot of zinc, it might feel metallic. I've never tried that. Actually, mice, they like small amounts of zinc. So if you, I'll give them a solution of water plus small amounts of zinc, they like the zinc solution. So it's actually not that aversive to them, I guess.
Right. Just because I know like with like those like lozenges that you take when you're like sick, the zinc lozenges, they say on the back that there's like a side effect where you can like lose your taste.
Honestly, I don't know about that. I have to look that up.
But
But
it's the higher doses, I think like 40.
probably higher doses and probably due to some other reason. But zinc is well known. It can regulate regeneration of skin, gut epithelial cells. So my theory is that it's probably doing something similar in the taste buds. But I don't know. I'd look that up. Whether you consume so much, you
Right.
lose yeah.
Okay, I think we'll call that good. Thank you. If you want to talk to Sunil later or afterwards, if you have more questions, feel free to ask.
So
And also remember, if you're interested in doing research in his lab, you should email him or tell me. But yeah, thank you, Sunil.
wonderful. Thank you.
No?
Ready to